This is the third out of the 5 sessions. So in the first session, I was giving a recap on cancer biology and the sorts of mutations that cancer cells contain and their impact. In the second session, I looked at I'm now forgetting targeted therapies and their mechanism of action. In this session, I'm introducing immunotherapy and beginning to talk about immune checkpoint inhibitors. And then in week 4, I will continue on that theme to begin with, looking at how we are trying to improve and build upon where we're at with checkpoint inhibitors so far. But I'll also look at some novel immunotherapies. And then the 5th session is about precision cancer medicine, which tries to bring everything together and use all our knowledge and all our treatments to help as many cancer patients as possible. So I will just sort out my slides on my screen and we will get started. Some reason my computer seems to have frozen, so I might try that again. I'll stop sharing and reshare, and hopefully my computer will play ball. Oops. Oh dear, what's it doing? Just try that again. Just 1 second. My computer is having a wee moment. Okay, all working now. Great. So in this presentation, I will introduce immunotherapy by first of all giving a bit of a recap on a very rough recap on the immune system as a whole, but then looking at how it is our immune system has a chance even to recognize and destroy cancer cells and how that happens. And I will be introducing the T cells as being the most powerful and potent and likely cell to begin killing lots and lots of cancer cells. They're highly equipped to do that. And a lot of immunotherapies are focused on those T cells. How do we help the T cells? How do we give them a boost? How do we leverage them? How do we persuade them to kill cancer cells? And I'll talk about the big picture of all the immunotherapies that target the T cells and then look at the immune checkpoint inhibitors as an important example of an immunotherapy that aims to leverage the T cells. So I'll begin with a very broad brushstroke introduction to the immune system. I hope it doesn't come as a surprise to you that there are many different types of white blood cell that exist in our body that have functions in terms of ridding our body of all sorts of bacterial infections, viral infections, parasitic infections. They keep our tissues and organs free of debris, free of damaged and dying cells, and importantly, also free of cancer cells. The white blood cells towards the left of this diagram, so all our white blood cells, well, there's never an all in biology, most of our white blood cells are produced by the stem cells in our bone marrow. Those stem cells self-renew, they produce more stem cells, but they also produce all these different types of white blood cells that mature and specialize and move into place and perform specific functions. On the left-hand side of the diagram, sort of roughly within the dotted red line, are the myeloid white blood cells. These are responsible for the very immediate response to any infection. They are precise in that they know precisely what it is that defines a bacteria, a virus, a parasite. They know what those look like. They know that they need to react to them. They know that they shouldn't react to healthy cells of the body, but they're not as precise as the lymphoid white blood cells, typically the B cells and the T cells. So the myeloid white blood cells, they know what a virus looks like, they know what a bacteria looks like, and so on. But they are not pinpoint precise to every specific type of bacteria or virus in the way that the B cells and T cells are. That means whenever we're infected by something, we might only have 30, 40, 50 B cells that recognize that thing or T cells that recognize that thing, because what our B cells and T cells are amazing at is diversity. Each B cell, each T cell is slightly different from every other, meaning that amongst all those billions of B cells and billions of T cells, whatever we are infected by, there are a handful of them that can respond to that infection. That's how our immune system tackles the problem that it does not know what we might be infected by during our lifetime. It just produces this massive diversity of B cells, of T cells, so that whatever it is we come across, hopefully there are some B cells and some T cells that can react to it, but it's going to take a few days for them to multiply up to sufficient numbers that they can really combat that infection and overcome it. So the B cells and T cells, at the start of an infection, there's only a handful of them that recognize the target, but they quickly multiply up, and then they can form an effective machine getting rid of that infection. The myeloid white blood cells are not as precise, not as specialized, so they can hold the fort in the meantime. They can mount an attack, try and keep everything under control until there are sufficient B cells and T cells to completely eradicate the problem. So you have these roughly two parts of the immune system, the myeloid side of the immune system and the lymphoid side of the immune system. Okay, so now I'm going to look at how it is that our immune system has even the potential to recognize and destroy cancer cells. And I'm going to start right back at the beginning of where I began in session 1. And I'll begin here with the fact that our cells contain these structures that we call the chromosomes. Those chromosomes are made from this substance called DNA, and within the DNA, there are stretches of the DNA that the cell can recognize as being genes. The instructions telling the cell how to manufacture all the many thousands of proteins it needs to perform all the many functions the cell needs to perform in order to keep us alive. So the DNA is the source of information, the source of instructions. It's the proteins that do all the work of the cell. The little machines for making proteins are known as ribosomes. And there's roughly 2 to 5 million ribosomes in every single cell. Those ribosomes are churning out proteins. So the cell is constantly making new proteins. It shuffles them around inside the cell so that those proteins are right where they're needed to perform whatever function it is they need to perform. But proteins wear out, they get old, maybe they weren't assembled properly when they were first made. Our cells need a means of recycling unwanted proteins, proteins that are in the wrong place, proteins that have gradually unraveled and can no longer do their job, proteins the cell no longer wants. The machinery for getting rid of proteins, that's the proteasomes. So our cells contain between 2 and 5 million ribosomes and another 1 million proteasomes. And any protein the cell needs to recycle and destroy, it feeds into the proteasomes. And as proteins pass through the inner core of the proteasomes, they're gradually chopped up and recycled. Now, not every protein that gets forced into the proteasome gets completely destroyed. Some of the fragments are larger that come out of those proteasomes, and some of those fragments the cell purposely puts into a system involving the Golgi apparatus, the endoplasmic reticulum, and it deliberately refines and trims and selects those little protein fragments for display on the cell surface. And in order to display all these little fragments on its surface, it produces tiny protein cups. And these tiny protein cups are called the MHC proteins. So when a cell is recycling proteins and destroying them, some of the fragments get trimmed to the right length, get inserted inside these protein cups, and get moved to the cell surface. The whole point of this is that passing white blood cells have an immediate opportunity to see exactly what is going on inside every cell in your body. White blood cells can't peer inside a cell. What they can do is scan its surface. So every cell on its surface is displaying all these little cups with tiny little fragments taken from every single protein it is making. And in this way, each cell in our body tells our immune system what's going on inside it. We call these little fragments peptide antigens. Peptide means a fragment of a protein. An antigen is anything our immune system might possibly react to. So these are peptide antigens presented to passing white blood cells so that those passing white blood cells know what is going on inside each cell. This system works really well for virus infections because any virus that infects a cell immediately injects its own DNA or RNA into that cell, forcing the cell to make virus proteins, and that's how the virus replicates. If a cell is making virus proteins, they will get chopped up by proteasomes. They will end up displayed by the MHC cups. They will be spotted by passing white blood cells, and the passing white blood cells will immediately see this is a cell that's been infected and sabotaged by a virus, and it will destroy it. Cancer cells, slightly differently, cancer cells contain mutations in their DNA. Because cancer cells contain their mutations in their DNA, they're making mutated proteins. Those mutated proteins get recycled through the proteasome system, just like all the other cells' proteins. Tiny fragments of those mutated proteins might end up displayed inside the MHC cups. Because these protein fragments from mutated proteins that only exist because this is a cancer cell with mutations in its DNA, because these are new antigens that should never normally exist, that only exist because of the cell's mutations, we call them neoantigens. So these are new antigens that come from mutated parts of proteins. And this is what our T cells can react to. So this is how our T cells have the possibility of killing cancer cells. Because if a T cell receptors on their surface match the shape of a cancer cell's neoantigens, that T cell will kill the cancer cell. So I'm going to say more about how this works in a second because it's important in terms of understanding how checkpoint immunotherapy works and sometimes doesn't work. So I will come back to this in a second. For now, I'm just going to say that neoantigens are what the T cells of our immune system can react to. Those T cells aren't the only sort of white blood cell that can react to and destroy cancer cells. The macrophages of our immune system can also kill cancer cells because one of the jobs of a macrophage is to look for and destroy any cell that looks damaged, that looks like it really ought to be dying, even if it maybe can't quite die. It's looking for signs of sickness, signs for abnormality, signs of stress, signs that things are going wrong. So macrophages are constantly destroying damaged cells and recycling them, and that will include cancer cells. And the natural killer cells, they can also kill cancer cells because they're looking out for any cell that has lost its MHC proteins. And that commonly happens in cancer cells. Is one of the ways cancer cells can avoid destruction by T cells is by just removing those MHC cups from their surface so that they're not displaying any neoantigens at all. The natural killer cells, they're on the lookout for any cell that has completely removed its MHC cups because that's a telltale sign of cancer, it's a telltale sign of virus infections, and so natural killer cells try to step into that gap and destroy things that T cells aren't going to recognize. Now, tumors commonly contain a lot of white blood cells, but exactly what those white blood cells are and what they're doing tends to change over time. What scientists believe is happening is that right at the early stages when cancer cells first emerge, a lot of those cancer cells will be detected and destroyed by the immune system. They'll look a bit sickly and get destroyed by macrophages. They'll have lost their MHC cups and be destroyed by natural killer cells, or they'll display neoantigens and they'll get destroyed by cytotoxic T cells, the cancer-killing T cells. So there might well be a long period where there's very little multiplication of cancer cells. There's very little increase in cancer cell numbers because the immune system is doing really well at keeping everything in check and destroying cancer cells as they emerge. But possibly years later, the cancer cells start getting the upper hand. At this point, the most visible cancer cells have been destroyed by the immune system, and the ones that are still surviving and multiplying are the ones that have found a balance. Maybe they've lost most of their MHC proteins, but not all of them. Maybe they've lost the signals that would tell the macrophages that they're sickly. Maybe they're managing to avoid destruction by the T cells. And a lot of the time, what has happened is the cancer cells have lost most of their MHC proteins, or the MHC proteins just aren't in operation properly. The neoantigens aren't getting slotted into place inside the MHC proteins, and so the MHC proteins are empty. And there's a significant proportion of cancers where the sorts of mutations the cancer cells contain just don't really lend themselves to the creation of neoantigens. If cancer cells contain relatively few but large-scale mutations, you're not going to get many neoantigens. What seems to be the best in terms of that cancer cell being visible to the immune system and displaying lots of neoantigens is when those cancer cells contain lots and lots of tiny mutations. If you think of a skin cancer caused by UV light from the sun, that cancer's DNA contains many, many thousands of mutations, is producing many, many different faulty proteins, and there's lots of opportunity for neoantigens to be created and slotted into those MHC cups on their surface. Likewise, a lung cancer in somebody with a history of smoking. The cigarette smoke has caused thousands of small scale mutations in the DNA. So any cancer where you get thousands of small scale mutations, that cancer is going to be more visible to the immune system because there's going to be more neoantigens. So scientists think that step one is a period where there's very, very little growth of the cancer at all. You've just got most of the cancer cells being destroyed and just a few surviving. Then there's a stage of equilibrium where the cancer cells are gaining the upper hand. And finally, a stage we call escape, where the cancer cells are continuing to multiply, where the white blood cells found inside the tumor are no longer cancer-killing white blood cells, but they're very often cancer cell protecting, cancer cell nourishing, and white blood cells that actually suppress T cells. The combination of white blood cells you find inside a tumor and their behavior differs a lot from cancer to cancer. It differs with the stage of at which the person is being diagnosed. The early stage cancers tend to have more cancer killing white blood cells, more kind of lively T cells able to kill a cancer cell. Metastatic cancer has often taken many years to get to that stage. There's been lots of opportunity for that tumor environment to change and to now be dominated by cancer protecting and T cell suppressing white blood cells. So you can very roughly categorize the sorts of white blood cells you find inside tumors as to whether they are good guys, whether they're killing cancer cells, or whether they are bad guys and actually protecting and supporting the cancer cells. So the good guys, these are the cytotoxic T cells, the T cells that can directly kill cancer cells. Natural killer cells, mature dendritic cells, helper T cells, often even B cells. These are helpful in terms of generating a cancer-fighting immune response. The bad guys include the regulatory T cells and the myeloid-derived suppressor cells. These actively suppress cancer-killing T cells, but they also include the tumor-associated macrophages, neutrophils, mast cells. These might initially have been able to kill cancer cells, but if they're hanging around in that tumor environment too long, they lose that cancer killing ability and they actually start supporting, protecting, giving energy, giving support signals to cancer cells and keeping them alive. There's always nuance. It depends a bit exactly what cancer it is, what stage it is. It even depends, varies according to the individual. But these are our themes that I think are a useful starting point in understanding how this works. And I wanted to compare and contrast pancreatic cancer with lung cancer. Pancreatic cancer is notoriously very, very difficult to treat successfully, and being diagnosed with it usually means that you are not going to live more than a year or two past that diagnosis. There are many reasons why pancreatic cancer is so difficult to treat. Some of it is to do with how fibrous and compacted the tumor is and how little opportunity there is for drugs to penetrate through the tumor. Some of it is to do with the mutations the cancer cells contain and what drives them, and the fact that we can't or haven't yet developed targeted treatments for this kind of cancer. But it's also to do with the types of white blood cells you find inside pancreatic tumors. You generally find all the bad guys, the white blood cells that are actively supporting, encouraging, protecting the cancer cells and suppressing any cancer-fighting T cells that might come along. So you find lots of myeloid-derived suppressor cells, lots of regulatory T cells, lots of tumor-associated macrophages, lots of mast cells. In contrast, inside a lung cancer, you will still find some of those bad guys, but you're much more likely to find also lots of good guys, useful natural killer cells, cancer-killing macrophages, cancer-killing cytotoxic T cells. What you can even find are tertiary lymphoid structures. These are pockets of tissue within the cancer where you've got lots and lots of white blood cells in that little tertiary lymphoid structure. And crucially, the types of white blood cell there are, the way they are arranged inside that structure, and the way they're talking to one another and communicating with one another, it looks a lot like what goes on inside a lymph node. And you get cancer-fighting T cells created from within that tertiary lymphoid structure. And those T cells really then don't have very far to travel at all before they're in amongst the cancer cells and killing them. So the presence of tertiary lymphoid structures is often a really good sign for the patient. Oh, apologies for my dog barking in the background. So T cells have the greatest capacity out of all our white blood cells to identify and destroy cancer cells. And the T cells, let me just shut the door behind me 1 second. So the T cells are one of these lymphoid type white blood cells. There are many types of T cell. I can't possibly go through all the different types of T cell there are. There's probably 30 odd. I don't know now. They're constantly finding more subclasses of T cells. But for our purposes, there are the regulatory T cells, which are immune suppressing. There are the helper T cells that are there to coordinate immune responses. And there are the cytotoxic T cells that can physically connect with cancer cells and destroy them. And cytotoxic T cells have this capability. They can get inside tumors, they can physically connect with cancer cells and kill them. There are many different forms of immunotherapy that have been developed where the target of that immunotherapy is to harness the powers of T cells and use them to kill as many cancer cells as possible. It's only gone a bit fuzzy, I think. Anyway. In this diagram, I want to say a little bit more about how the T cells become activated, because this is all building up to understanding how immune checkpoint inhibitors work. So T cells don't patrol the body looking for cancer cells. That would be a very inefficient way of using our T cells. As I say, we've got this massive diversity of T cells in our body. Only a few of them can recognize any individual target. So you don't want to send them all out looking for that target. They probably never find them. Instead, our T cells tend to congregate in the lymph nodes. And when they're in lymph nodes, that's when they communicate with the dendritic cells. And the dendritic cells are the body's patrolmen. The dendritic cells are out and about in organs and tissues, looking for signs of problem, looking for signs of damage, infection, looking for signs of cancer cells. And when they find anything that looks a bit dodgy, they start hoovering up all the proteins or the debris they can find in that environment. They process it, they display it on their surface in their MHC cups, and they report back to the T cells and lymph nodes. And they start showing the T cells everything that they found. And if they find a T cell whose T cell receptors match the shape of one of the peptide antigens displayed by the dendritic cell, that T cell is part way to becoming activated. The T cell needs a second activation signal. It needs its CD28 protein connecting with a B7 protein. But should those two connections take place, and should the T cell also receive extra signals from other white blood cells, it will go into activation mode. It will multiply very quickly, and it will become a fully fledged activated T cell. So if we look at how this works inside a tumor now, inside that tumor environment, there's probably a lot of dead and dying cancer cells. This is a feature of the tumor environment. It's low in oxygen, it's acidic, it's a very sickly and hostile environment. There are cancer cells multiplying, but there are also cancer cells dying in. There's plenty of debris. Hopefully, that debris sends an alarm signal to any dendritic cells that find their way into the tumor. And hopefully what happens is the dendritic cells enter into that environment. They realize that this is an unhealthy environment, and they start picking up all the debris they can find, all the proteins that have spilled out of the dead and dying cancer cells. And when they're loaded up with all that debris, they take it perhaps to a tertiary lymphoid structure or a nearby lymph node. And as they travel to that lymph node or that tertiary lymphoid structure, they internalize, they process all that debris, they chop it up into the right size fragments, and they put it inside their MHC cups so that by the time they reach the lymph node or the tertiary lymphoid structure, maybe 24 hours later, by the time they reach the T cells, they're displaying lots of fragments from all the debris they picked up, and they show it to the cytotoxic T cells. And they're looking for cytotoxic T cells whose T cell receptor matches a piece of debris showed to it by the dendritic cell. And hopefully that piece of debris is a neoantigen. It's a fragment of a faulty protein that is being produced by the person's cancer cells, so that if the T cell reacts to that neoantigen displayed by the dendritic cell, if it enters into the tumor and meets the exact same neoantigen, but now displayed by a cancer cell, it will kill that cancer cell. So it's the dendritic cell that picks up the debris. It takes it to the lymph node or the tertiary lymphoid structure. It shows the debris to T cells and activates them. And hopefully what the T cell is recognizing is a neoantigen. So that when it is activated and it multiplies and this new army of T cells enters into the tumor, they all recognize that same neoantigen, which is displayed by the person's cancer cells. However, tumors are not kind to T cells. One issue we have is that if the person's cancer cells don't contain many mutations and aren't producing many neoantigens, this system might never work at all. None of this might happen. If dendritic cells are picking up debris, but there's no neoantigens there, then you won't get T cells activated to then move back into that tumor environment. Also, even if suitable T cells are created against neoantigens, they might get trapped inside blood vessels. They might never find their way into the tumor environment in a way that brings them into close contact with cancer cells. In addition, in the tumor environment, often what you find is that the white blood cells there are producing signaling molecules which suppress cytotoxic T cells, or there's lots of myeloid-derived suppressor cells there, there's regulatory T cells there, and they suppress the cytotoxic T cells. Or sometimes the cytotoxic T cells just become exhausted and tired and suppressed because there's a lack of nutrients, there's a lack of oxygen, there's lots of toxic waste from dead and dying cancer cells that suppresses the T cells. And the T cells can also be suppressed via the checkpoint proteins on their surface, which of course is something I will come back to in a second. Lastly, even if the T cells recognize neoantigens, start killing lots of cancer cells, they can't keep doing that forever. They will run out of the toxic enzymes that they use to kill cancer cells. They become depleted, they become exhausted, and even if they try and keep killing cancer cells, they just can't do it. They've become worn out. So in an ideal world, what happens is that your immune system realizes that there's cancer cells present, dendritic cells pick up debris, they show the debris to the T cells become activated, they multiply, they find their way into the tumor environment, they start killing the cancer cells, they manage to stay not suppressed, they manage to stay active, and they kill so many cancer cells that they bring everything under control and completely eradicate all the cancer cells present. So that's your ideal scenario as to how your immune system would react if cancer cells emerge. But as I've said, not every cancer is visible to the immune system. You don't always get enough T cells created to completely eradicate your cancer cells. They may never get in amongst the cancer cells in order to kill them. They sometimes get trapped in blood vessels or trapped around the outskirts of a tumor. They might enter a tumor environment but quickly get suppressed by myeloid-derived suppressor cells, by regulatory T cells, etc. They might never reach the levels needed to keep the cancer under control. And unless they're being refreshed with new T cells, they're going to just burn out and become suppressed and never completely eradicate all your cancer cells. So the goal of immunotherapy is to help the immune system. And many immunotherapies, as I've said, the goal is to specifically help the cytotoxic T cells. And many immunotherapies have been developed with different mechanisms of action where the goal is to assist in a different part of this process. The vaccine-based immunotherapies, the goal there is to help the immune system recognize the fact that you have cancer. So you're feeding neoantigens into the body, hopefully somewhere that passing dendritic cells will pick them up. Or sometimes you're taking dendritic cells out of the body, purifying them, feeding them in neoantigens and putting them back again. Or we have things like the oncolytic viruses, where they infect cancer cells, create lots of new debris that hopefully the dendritic cells react to. So it's all about helping those dendritic cells recognize that you have cancer and help them pick up those neoantigens. There are then some immunotherapies where the goal is to increase the number of activated T cells created. There are ideas as to how we might help the cytotoxic T cells penetrate into the tumor environment. There is an idea and some evidence that angiogenesis inhibitors that target blood vessels can somewhat do this, although I'm not sure that's conclusive proof. There are various treatments where the goal is to help the T cells remain active for longer and to not get suppressed. Then there are things like tumor infiltrating lymphocyte therapy, where you take a piece of tumor, you isolate the T cells, you purify them, you activate them and put them back into the body. So you're trying to increase the number of cancer-fighting T cells that exist in the patient's body. And the T cell engagers, where you're trying to create a physical tether holding together cancer cells with T cells to encourage the T cells to kill cancer cells. So you're redirecting T cells. It doesn't matter what the natural target of the T cells is, you're redirecting the T cells towards a new target on cancer cells. But this does depend on there being some lively T cells in the tumor to begin with, and those don't always exist. And then finally, CAR-modified T cells, TCR engineered T cells. This is when you take T cells out of the blood, you genetically alter them so that they produce a new receptor protein, one that recognizes something on the person's cancer cells. This is an effective treatment for hematological cancers. It's much more of a struggle to make this work for the solid tumors. You might have created some really powerful cancer-fighting T cells, but they don't necessarily know where the cancer cells are to find them. And when they enter into a tumor environment, they can quickly become suppressed. So there's extra obstacles when it comes to creating a modified T cell therapy for a solid tumor. So out of all these different types of immunotherapy, I'm going to focus for the next 10 or 15 minutes on checkpoint inhibitor immunotherapy. And this is predominantly antibody-based treatments where the target is CTLA-4, PD-1, or PD-L1. With the CTLA-4 targeted antibodies, the effect that has is to increase the total number of activated T cells in the body. Any T cell sitting inside any lymph node is more likely to become active. The PD-1, the PD-L1 antibodies, here the target is T cells that have moved into organs and tissues. They're not in lymph nodes anymore, and you're making it more likely that they will stay active. I will begin by saying just a little bit more about T cells. T cells are incredibly powerful white blood cells. They kill cells infected by viruses. They control the immune system to a large extent and coordinate immune responses, and they destroy cancer cells. So they're incredibly useful white blood cells. The T cells that coordinate all the other white blood cells, that's the helper T cells. The T cells that directly kill virus infected cells and cancer cells, that's largely the cytotoxic T cells. Each T cell is creating a unique T cell receptor but it might have maybe 100,000 copies or so of that specific T cell receptor sticking out of its surface. T cells also have another 200 or so proteins sticking out from their surface, but the T cell receptor is the one that connects with a target. T cells are vitally important to us. They keep us free of viruses, they keep us free of cancer cells, but if the wrong T cell is activated at the wrong moment in time or remains active for too long, or becomes activated despite reacting to healthy cells rather than damaged or virus infected cell, that T cell is likely to do a lot of harm. T cells remaining active beyond what is useful causes chronic inflammation. T cells that react to something on healthy cells, those T cells are likely to cause an autoimmune problem. So the body needs a way of keeping T cells under tight control. And the means it has to do that are the checkpoint proteins found on the T cell surface. Some of those checkpoint proteins are inhibitory. If they're triggered, the T cell becomes less active, it might even die. Others are stimulatory. If they are triggered, the T cell becomes more active and more likely to stay alive. What is supposed to happen is that we get an infection, cancer cells emerge, whatever it is, T cells are activated to deal with that problem. They enter into the environment, wherever that problem is. Is it infected tissue? Is it a tumor environment? The T cells move into that environment, and as well as killing the target cells, they also start sending signals out into their environment saying, hi, I'm a really activated T cell. And at some point, my job will be finished. And at that point, I'm going to need help shutting down and dying off. So these activated T cells are sending out signals telling other cells in their vicinity to get ready for when the T cell's job is finished. And what those neighboring cells do is they start putting proteins on their surface that trigger the inhibitory proteins on T cells. So this T cell, I've given you an example, one of the inhibitory checkpoint proteins it has on its surface is PD-1, and cells in its close vicinity start putting PD-L1 on their surface in readiness for when that T cell's job is done, at which point this other cell comes in, it uses that PD-L1 to connect with PD-1 and it shuts the T cell down and it tells the T cell maybe to die. So this is a perfectly normal process. It's a healthy process. It is one that protects us from T cells remaining active beyond what is useful. It avoids chronic inflammation. It avoids some autoimmune problems. The fact that as soon as a very active T cell enters into an organ or a tissue, it tells the cells around it, start preparing for when my job is done. You will need to suppress me. The problem we have in a tumor environment, though, is not just white blood cells and other healthy cells putting that PD-L1 on their surface. Cancer cells also put PD-L1 on their surface, and they use it to shut down T cells that get too close. So often inside tumors, you find lots of cancer cells as well as other cell types with PD-L1 on their surface, and they're using it to shut down useful T cells. What our checkpoint inhibitors do is to keep the cells, to stop PD-L1 connecting with PD-1. So you're preventing that signal, that inhibitory signal, from being triggered. If you can prevent PD-L1 from connecting with PD-1, the T cell does not receive the instruction to become suppressed or die. Instead, it does the opposite. It stays alive and it stays activated. One thing always to bear in mind with these immune checkpoint inhibitors is that you can't control which T cells become activated. You hope that the T cells you activate are ones whose T cell receptors connect with neoantigens on the surface of the patient's cancer cells. These are the T cells that you want. These are the T cells you're hoping for. These are the T cells that you're praying that your checkpoint inhibitor keeps active. Because if this T cell receptor matches a neoantigen displayed by the person's cancer cells, then it will connect with that neoantigen and it will kill the cancer cell. And you will have done that person a lot of good if you've managed to activate sufficient numbers of T cells to really make a difference and keep that cancer under control. So the T cell receptor is connecting with the neoantigen. That's the signal for the T cell to kill the cancer cell, and you're keeping the T cell active with a checkpoint inhibitor, which prevents, in this case, PD-1 from connecting with PD-L1. But what you haven't done is help the immune system recognize that the person has cancer. You haven't created neoantigens for dendritic cells to pick up and take to T cells. You haven't done anything apart from boost the activities of cancer-fighting T cells that already existed prior to you giving that person the checkpoint inhibitor. If their cancer cells contain relatively few but large-scale mutations, and there are no neoantigens on the cancer cell surface, this approach won't work. So there's lots of people for which checkpoint inhibitor immunotherapy is not effective because their cancer cells have never led to the creation of cancer-fighting T cells. And you can't boost what doesn't exist. If there are no cancer-fighting T cells, then there's nothing for your checkpoint inhibitor to do that's useful. So there's lots of people who don't benefit from immune checkpoint inhibitors. Maybe their cancer cells are hidden from their immune system. There are no cancer-fighting T cells, or maybe there are cancer-fighting T cells, but they're being suppressed not via their checkpoint proteins, but by myeloid-derived suppressor cells, by regulatory T cells, by the cytokines in the tumor environment, by the lack of oxygen, by the acidity in that environment. As I mentioned, there's lots of reasons why T cells become suppressed inside tumors that have nothing to do with checkpoint inhibitors. So when you give a checkpoint inhibitor, you hope that their cancer is visible to their immune system. You hope that there are cancer-fighting T cells that are being suppressed via the checkpoint proteins on their surface. And it's only if those things are true that that treatment can help the patient. And sometimes you inadvertently boost the activities of T cells that don't attack cancer cells and instead attack healthy organs and tissues in the body. As I said, you've got no control over what T cells you activate with your immune checkpoint inhibitor, and you can inadvertently activate T cells whose target is healthy organs and tissues and not cancer cells. And now you've given them a boost and they cause damage and they cause inflammation and they cause autoimmune problems. The PD-1 and the PD-L1 antibodies are all doing the exact same thing. They're preventing the connection between PD-1 and the PD-L1, and they have an impact on T cells that have left lymph nodes and that are in organs and tissues. The CTLA-4 targeted checkpoint inhibitors are a bit different because T cells put CTLA-4 on their surface when still inside lymph nodes. So you're activating T cells wherever they are in the body, wherever lymph node they happen to be in. Another impact of CTLA-4 targeted antibodies is that you deplete the number of regulatory T cells. And what we don't know is which of those two impacts is more important. Do CTLA-4 targeted antibodies work because you're depleting regulatory T cells or because you're boosting the number of activated cytotoxic T cells, or maybe both? We don't know. This table lists most of the approved immune checkpoint inhibitors. There are constantly more entering into the marketplace and into clinical trials. Wherever there is an effective treatment created by one drug company, other drug companies want their own version because there's money to be made, there's clinical trials they can perform, they can find a niche for their treatment. So we have many different PD-1 and PD-L1 targeted antibodies, but they're essentially one treatment doing the same thing. If nivolumab hasn't helped a patient, avelumab isn't going to. It's essentially doing the same thing. Relatlimab is a bit different. It targets a checkpoint protein called LAG3. It, as yet, has only proven to be helpful against melanoma skin cancer. Ipilimumab and tremelimumab are two checkpoint inhibitors that target CTLA4. And as I've mentioned, the chief limitations of these immune checkpoint inhibitors are firstly, they don't work for the majority of the people they're given to, either because their cancer is invisible to their immune system and there are no cancer fighting T cells, or there are cancer fighting T cells, but they're being suppressed through mechanisms that don't involve the immune checkpoint proteins on their surface. Another limitation is the side effects they can cause. These are unpredictable in terms of their timing and their severity, and they can lead to lifelong problems. This is maybe not such an important consideration if you're giving checkpoint inhibitors to somebody with advanced cancer that would be lethal to them. Obviously, you need to kind of throw everything you've got at that cancer, and hopefully, if you've helped the person with the immune checkpoint inhibitor, it's a secondary issue that you may have inadvertently caused side effects. But if you're using the immune checkpoint inhibitor as a neoadjuvant or an adjuvant treatment for early cancer, where quite possibly that person is cured by their surgery, it's a much bigger deal to be adding in immune checkpoint inhibitors to patients who are potentially already cured, and you're possibly going to cause them a lifelong problem with a problem with their pituitary gland or their thyroid gland, or one of these autoimmune problems you can inadvertently cause with immune checkpoint inhibitors. If you want to improve response rates, if you want to improve the proportion of patients who benefit from an immune checkpoint inhibitor, then maybe you need to be more selective about who you give them to. Look for a sign that that checkpoint inhibitor is going to work, or you use that checkpoint inhibitor as part of a combination, possibly creating combinations where the one treatment enhances the effectiveness of the other, or more realistically, you're using two treatments in combination so that the patient doesn't miss out on the possible benefits from either treatment. Again, mitigating side effects is an important issue. How do we reduce the impact of the immune checkpoint inhibitors? How do we reduce the problems that they can cause? And again, another way to mitigate side effects is just to restrict the use of immune checkpoint inhibitors and make sure that we're selecting the patients who most need them. I'll finish with a quick summary. I began by saying that tumors are complicated places. You can find lots of different types of white blood cell inside tumors. Exactly what white blood cells you find and what they're doing there, it changes over time. Initially, long before the cancer is diagnosed, the white blood cells you find in there are likely to be ones that are cancer killing. The cytotoxic T cells, cancer killing macrophages, cancer killing natural killer cells. But as time goes by, that environment changes, and you're more likely to see macrophages that become part of the problem. They are supporting and nurturing of the cancer cells. The cytotoxic T cells, often they wear out, they become exhausted. If they're not being replenished, then they can't keep killing cancer cells. We also increasingly in metastatic disease and advanced disease, we see more and more myeloid-derived suppressor cells accumulating, Tregs accumulating, and they actively suppress cancer-fighting T cells. The goal of most immunotherapies, particularly ones for solid tumors, is to help the T cells. How do we help those T cells recognize cancer cells? How do we help them stay active for longer? How do we boost their numbers? This is the goal of many different forms of immunotherapy. The immune checkpoint inhibitors, they have one specific mechanism of action. They prevent the suppression of T cells by preventing the triggering of inhibitory checkpoint proteins on their surface. But as I said, they will only work if the patient's immune system has already realized they have cancer and has already created cancer-fighting T cells for you to boost. If those T cells don't exist, then a checkpoint inhibitor is not going to work. And when I come back next time, I will talk about biomarkers. How do we predict who is the most likely to benefit from a checkpoint inhibitor so we can be more selective in their use, and what combinations have been created that try and boost response rates. And I will also next time look at some of the newer forms of immunotherapy and how they are designed to work. In the meantime, I will stop sharing. I keep looking at the little image of myself and thinking I've gone really fuzzy. So apologies for that.
But it didn't
No,
detract
it's just
from
sort of
the.
distracting. We're looking at this sort of blurred image of yourself. I think sometimes the webcam has problems when it's a bit gloomy.